and she did her MSc degree in IIT Delhi during the year 2005 to 2007. And after his MSc degree, she joined the group of Professor V. Ramamurthy at University of Miami, USA. Then after finishing his PhD degree, she had carried out his postdoctoral studies with Professor Christopher Alabi and Professor Jinju Jiu at Cornell University and Columbia University. And one important highlight is that she had a big impact score of H index 23 and I-10 index 34 and overall 1973 citations for her around 60 publications.
Yeah.
Then she had won many awards and recognitions, especially that Outstanding Woman Researcher in Chemistry Award in the 9th Venus International Women's Awards. And she had many patents and book chapters to her credit. So with this brief introduction, I welcome and request Professor Mintu Porel to deliver the talk. Thank you.
Thank you, sir, for the kind introduction. And my sincere thanks to all the organizers and Professor T. Pradeep, sir, for the invitation, for giving me the opportunity to discuss our work here. So today I'll talk about some of the recent activities in our lab on the rational design and synthesis of macromolecules with structural tunabilities for environmental applications, a custom-made approach. So let me first thank them who are all behind all the works. I'm just presenting, but they are the main people behind all the works. So thanks to my current group members and all the alumni for their hard work, and all the collaborators for their intellectual participations, all the funding agencies for the financial support. So I would like to start the presentation with a great quote by Professor George Hammond. In 1968, in the Norris Award lecture, what he mentioned is that the most fundamental and lasting objective in synthesis is not production of new compound, but production of properties. So that is the motivation of our work. So what we are trying to do, we are trying to basically design and synthesize the organic macromolecules or materials where we can bring new properties as well as we can tune the properties to make them suitable for different applications. So currently we are working on the different classes of macromolecules. For example, sequence-defined polymer that I'll discuss in the next slide, conventional polymers with different architectures, macrocycles, and also the small molecular conjugate. So in all the platforms, our main idea is that we are incorporating different functional groups. Those different color codes are for different functional groups. And via different functional groups, how we can tune the properties for different applications. And in the application side, we are mainly exploring to develop the sensor as well as removal systems for environmental toxic pollutants. And in the biological sides, we are exploring how we are developing the new antibacterial drugs or to understand what are the factors that are important for interacting the macromolecules with different proteins. So let's start with the first one, the sequence-defined polymer. So what is sequence-defined polymer? The sequence-defined polymers are the polymers in which the monomers, they are arranged in a precisely sequenced manner. Very well-known examples are like peptide. We all know the peptide is a polymer, right? So here the monomers are 20 different amino acids. Now the sequence of different amino acids are crucial for bringing different properties and functions of the peptides. Now those are the natural polymers, right? But if we see the synthetic polymers, so majorly we have the homopolymers or the copolymers where we have either one or two kinds of the monomers, and those polymers, as we all know, that they are so important for different applications. Now combining these two systems like natural and the synthetic, if we can develop a synthetic but sequence defined polymer, then we can add many advantages. Like via chemical synthesis, we can bring massive chemical and structural diversity In that way, we can basically tune the properties thoroughly for making them suitable or best fit for different applications. Now, motivated by that, we have developed a novel class of sequence defined polymer, or SDP as a short form, and in our SDP we have strategically incorporated a key functional group, that is dithiocarbamate. The rationale behind selecting the dithiocarbamate group is that it was already well known in the literature that group has lot of basically applications in the material as well as biological fields. So now whatever the synthetic strategy we have developed for SDP, that is based on a key monomer that we have designed and synthesized in the lab, that is we call it as amine hydroxyl monomer. So here is the amine group and it's the hydroxyl group, and based on this monomer we have selected a commercially available co-monomer chloroacetyl chloride. So how the reaction goes, this amine in presence of carbon disulfide reacts with the chloro group to form this dithiocarbamate group. On the other hand, that hydroxyl reacts with the acyl chloro group to form the ester bond. And now through this R group, we are incorporating a wide range of the functional groups. And this is the two-step synthetic strategy that we have developed. It's very simple synthetic strategy, and it is tolerable with a wide functional group range. Now, let us see how we are developing the SDP, or sequence defined polymer. So, we start from here, then in the first step we add carbon disulfide and the first monomer, then come to this step. And then next the co-monomer comes, and then we come to here where R1 and R2 are the two tunable functional group centers. Like that we can do the n number of the cycles, and at the end we can get the SDP with our desired n marks or desired chain length. It is a fixed chain length, control chain length, and these different color codes represent the different functional groups. And also there are some advantages of this strategy. So, we have optimized those two steps in such a way that it gives complete 100 quantitative yield or 100% yield in very short time. First is the 30 minutes and second one is the 20 minutes. And also the reaction conditions is completely mild reaction conditions. And also another thing is that although we are doing the sequential addition of the monomers, but this strategy is completely support free as well as the protection chemistry free. So, this is a very simple strategy, and also it is a mild reaction condition from inexpensive starting material. So, now generally after we do the synthesis, we thoroughly characterize by proton and C13 NMR as well as mass spectrometry. And now I will show that one. As I mentioned, the hydrophobicity can be judged by high performance liquid chromatography, or HPLC, in which we know that the x axis is the retention time. When it goes from lower to higher means it's getting more hydrophobic. And we can see here that we have a nice window from different SDOs with different hydrophobicity. So, and also this synthetic strategy, it is basically very scalable. We can make the multi-gram production of SDPs in a very short time, and also we advanced our synthetic strategy to make the post-synthetic modification. After we synthesize our SDO, we can bring that secondary systems via post-synthetic modification via different routes. That's also we have established. So, after we establish this SDO synthetic strategy, we are now exploring different applications, and today due to the time limitations, from the material side I will show that how we are developing the sensor as well as removal of toxic pollutants, and the biological sides I will show about the antibacterial drugs and the protein interactions. So, now in the literature, it was already known that mercury has a very good interactions with dithiocarbamate, and that property we have utilized for developing the mercury sensor as well as removal system. So, as we all know that mercury is very toxic, right. So, from different industries it is polluting the water, and through water bodies it is coming to the human bodies, causing lot of basically diseases. Even few years back the Minamata disease, it was all for the mercury poisons, and that took away a lot of lives. So, it's very important to develop the mercury sensor as well as removal systems. So, what we did for that, we attached a fluorophore, that is a dancyl fluorophore, with our SDO system synthetically. Now, this dancyl tag SDO, when it is free, it is yellow in color, and when it comes in contact with mercury, its fluorescence is completely quenched. You can see here, and that's what we are using it for the sensing. So now for sensor, two things are very important. One is selectivity, another was sensitivity. So what we found that this SDO, the Dansile Tact SDO systems, it is very selective to mercury in presence of all the metals. We can see from the fluorescence it's only quenched in presence of mercury. We can see also we can have the visual detection as well. We can see here. So it is highly selective, that's what we proved. Next is the sensitivity, that means how low level we can detect the mercury. And here comes one advantage of our SDO based platform than the conventional sensor platform. So here, as it is a sequential addition, basically it is a oligomeric systems, we can increase the number of dithiocarbamate unit, right. So what we observe, when we increase the number of dithiocarbamate units, its sensitivity is improving. You can see here, when N is the dithiocarbamate group, when it is 1, it can detect up to nanomolar level. When we increased N dithiocarbamate group to 2, it can detect at the picomolar level as well. So it is very sensitive. So after that, basically after confirming that, we also have developed a paper strip based sensor. So it's a sensor coated paper strip, and when we add our mercury, we can see it's fully quenched. We also have tested in the real water sample, that is lake and tap water in our campus, and that also works well. So now after confirming that it can detect very low level of mercury in water, we have also studied that whether it can detect in the living cell as well. So here we can see these are the cells, and this is the cell in presence of our sensor, and we can see it is blue in color. So that says two information. One is that our sensor is cell permeable, and we can also use it as a imaging purpose as well. And then when we add mercury, it says that fluorescence gets completely quenched. So it proves basically that our sensor, basically it can detect mercury in the water as well as living cell. So now after we confirm it is that it works as it can detect mercury, our next goal is that how can we remove the pollutant from water, right. So that is the next one. So when we started that work, then of course that we all know that in water not only the mercury or the heavy metals, there are many other pollutants, for example, pharmaceuticals, agrochemicals, organic dyes, feed additives, etc. So what we thought that instead of making a single material for a single class of individual material for individual class of pollutants, can we develop a general polymeric platform or material platform in which via tuning the functional groups, we can target the multiple classes of pollutants. So that's what basically the goal in this one. So with that, what we develop, again the dithiocarbamate based organic polymer, that's what we call DTCOP, dithiocarbamate based organic polymer, in which via tuning the functional groups we are able to remove different classes of the pollutants, actually multiple classes of the pollutants. So today, due to the time limitations, basically I will show few examples here. So this one so this is the common structure, that's what we designed. Here this is the structure of the polymers, and you can see this is the dithiocarbamate, and through this R1 and R2 we can tune the functional groups in our polymers. And the synthetic strategy, this is a two-step synthetic strategy. This is also in very 1 hour we can make multi-gram productions of this polymer from a very inexpensive starting materials, and we can tune its functional groups. As I mentioned that functional group has a influence on its property. Just one simple example you can see here, that these two polymers, in both the cases actually R1 is the same, but R2 one case aromatic and other case aliphatic. We can see one case it is a powder, other case we find the glassy solids. So it says basically the functional group has a huge role in tuning its property. And this dithiocarbamate based organic polymer shows that it is very efficient to remove mercury. So it shows almost complete removal of the mercury. From ICP analysis we found complete removal of mercury in a wide range of the pollutions, basically 5 to 400 ppm range, with the uptake capacity of 413.7 milligram per gram. So now when we think of the practical applications, recyclability of the material is very important, and we tested up to 3 cycles, and it shows basically very good recyclability. And another crucial point is that when it will remove the toxic mercury, it should not touch the other essential metals, and that's what also we have tested, that while it is completely removing mercury, it does not touch that sodium, potassium, calcium, those kind of essential metals as well. And it works in a wide pH range from pH 4 to 11, and also we have tested with the real water samples, that lake and tap water in our campus, and it works really well. So now after confirming that, what we developed is spin column technique in the lab. So it is a small mini column, and then we load our material, then we add mercury polluted water, and then we spin it at a high spin, and by that what we found, in just 30 seconds we can get the complete removal of mercury. So now if we combine those two systems, that sensor as well as removal, we have developed a detection and detoxification toolkit for mercury. When we get the contaminated water, through our sensor we can first test that whether mercury is present or not, and if it is present with our material, we can remove the mercury as well. So after mercury, as I said, that we were testing different other pollutants. So similarly, I will show another common pollutant is the dyes or the colors, because from different textile industries or paint industries, even from our household like washing of our clothes, all the dyes or colors are coming and contaminating water. And many of the dyes, they are aromatic enriched. So what we did in our polymer, we have made basically that we increase the aromatic sites in the R1 and R2 positions, and that shows that complete removal of dyes or colors, multiple dyes basically, like you can see that Congo red, Eriochrome bacteria, some common dyes we have tested, methyl orange, malachite green, crystal violet. So all the cases basically it shows complete removal. So this is actually before it is the dye absorption, and after treating with a material, it is completely off. So after testing the individual dyes, what we did, we just mixed all the dyes together. What is the practical case actually, and in that case also it was able to remove the dye completely. So two things basically, I will show one video that what we can see, that it is efficiently removing and it's very fast. So here you can see this is our materials, and when we add the colored water, you can see immediately when it comes in contact with material, whatever water is coming, it is completely colorless, and when we don't have the materials, it's a control, it's a full color. So it basically shows that it is very fast and very efficient, efficiently it is removing the different dyes. So like that, when we are exploring different other classes of the pollutants in water, then we thought that we will let us try our materials for the air, some pollutants in air, and then we found that iodine is a common pollutant coming from nuclear plant house. It is contaminating in air as well as water and air as well. And iodine is a Lewis acidic in nature. In our material, we have a lot of Lewis basic centers, and through Lewis acid basic interactions, our materials was able to remove iodine from air and water as well. So you can see this, the material in the iodine vapor, it's completely black colored, and it was well characterized by Raman spectra as well. And then when we wash with ethanol, iodine is stored in ethanol, and then we can recycle back the materials. And then it was also able to remove iodine from water as well. So now like that, so due to the time, I will not go into the other classes of the pollutants, but overall what we can say that we have developed some kind of multi-purpose adsorbent material which is able to remove different pollutants from water as well as air, air as well. So now I will show basically, as I said, that what we with this material we made a mini column chromatographic technique in the laboratory scale. So where it shows that the single platform, material platform is removing different basically dyes, and as well as, so it is a kind of Congo red. We can see that this is a small column, we have the material, when it immediately basically it's coming like water is free from the dyes. This is another color, is that Eriochrome black tea, same thing it is happening. So when it is passing through our materials, the water is coming completely colorless, and this is another, it's not dye, it's the iodine. So here also you can see it is coming basically colorless. So all those cases, so one important thing is that not only it has to be efficiently removed, it has to be very fast for that column chromatographic technique, and this is actually removing, everything is very fast. So like that, basically our SDO systems, we are exploring different classes of material applications. So in the next session, last few minutes, I will spend that some, I will show some examples how we are employing our synthetic systems for different biological applications. So let us start with the antibacterial drug development. So here what was our hypothesis? What we found that sequence defined oligomers platform is a very potential platform for antibacterial drug agent development. So what we know that in the bacterial cell membranes, it has a negatively charged phosphate group and the hydrophobic lipid moieties. So what we did synthetically in our SDO system, we added the cationic charge and the hydrophobic units, so that the cation and electrostatic interactions and the hydrophobic interactions, our SDOs will come into contact with the cell membranes, then disrupts the membranes towards the cell death. So that was basically our design, and with that I will show a very simple design of our SDO system, that again dithiocarbamate based systems. It's just simple 2-mer, and in only one center, that R group, we have systematically increased the hydrophobic groups like aliphatic chain length from C3 to C10, all systematically we have increased the aliphatic chain length, then cyclic aliphatic systems as well as some aromatic groups as well. And so after doing the synthesis and characterizations, as hydrophobicity is main important factor here, we have tested the hydrophobicity from HPLC, and it shows it has a very nice window from hydrophilic to hydrophobic characters. So after that, we have basically tested this library for the antibacterial activity for 2 types of the bacteria, gram positive and gram negative. So gram negative E. coli and gram positive Bacillus subtilis. So here we are measuring the MIC value, that is the minimum inhibitory concentration to kill the bacteria, right. So MIC value is lower, means it's good, it's more active. So when we test it, what we can see, when we are increasing the chain length systematically, with increasing the chain length, we can see the MIC value is decreasing, and we found like 3 microgram per ml. This is a really good number basically for the antibacterial activity. Like that, basically through our this structure activity relationship, we have screened the compounds which shows antibacterial activity. So next is that it has to be, important thing is that it should not have any cytotoxicity. That we have tested by the hemolysis assay, and that also shows, confirm that it has a very negligible cytotoxicity. So like that, through the synthetic system, that's SDO based platform and synthetic systems, via carefully systematic structure activity relationship, we found a candidate which shows a very good antibacterial activity and negligible cytotoxicity. So after confirming, we further went for the mechanism, thoroughly characterized the mechanism of it via 2 systems. One is the scanning electron microscopy, another one is the GUV. So first is the scanning electron microscopy, where we can see that the bacteria, it is a nice rod shaped bacteria, and after treating with our best compound, the DEC compound, we can see its cell membranes is completely disrupted. So this is a pure like confirmation that yes, it is a membrane targeting activity. And also we have tested the same mechanisms via GUV, giant unilamellar vesicles, or the GUVs, that mimics the bacterial cell membranes. And actually these are the vesicles, the GUVs, these mimics the bacterial cell membrane, and when the GUVs were treated with our SDO, decal SDO, so this we can see the disappearance of the GUVs, and that disappearance of the GUV prove that its membrane is completely disrupted. So through those 2 systems, we are also probing the mechanisms that whatever we have hypothesized, how the design was completely based on the cell disrupting mechanism. So it proves, at the end it proves also that. So like that, in this project we are also basically going through the doing the other synthesis and other libraries for more and more antibacterial works. In another project, we are also trying to understand what are the factors in the synthetic macromolecules are important to tune its interactions with different proteins. So for one example is that what we have done, that we have actually developed a library of the macromolecules via changing different factors, functional group, chain length as well as architectures, and at the end we are interacting each one of them with the sets of the proteins via fluorescence resonance energy transfer or FRET. And from this thorough study, experimental and theory, we found one candidate which is selectively interacting with the serum protein, and based on that, as it was fluorescent level, based on that we developed a serum selective staining agent, so which is useful to basically detect the serum, because serum albumin is important for different diseases. So that also we are basically currently exploring that factors as well. So in this basically presentation, so I mainly focused on one functional group, as I have shown so far, the dithiocarbamate based functional groups, and 2 systems, that sequence different polymer as well as the conventional polymer. So last few minutes, I will just mention that we are also, like the dithiocarbamate group, we are also exploring different other functional groups as well as architectures. So one example is that recently we developed working on the amine group as well, because amine is also basically is an important functional group for many biological applications and also material applications as well. So very recently we have developed another novel class of the sequence different polymer based on completely amine. So this is the amine based sequence defined polymer. So that already we have established synthetic strategy and we are exploring its application as well. And not only the sequence defined polymer, we also are developing the amine based conventional polymers, in which we are, by tuning the functional groups, we are tuning its properties for various applications. So like the functional groups, another key, because our main idea is to tune the properties. So functional group is one key factor. Another factor is the architectures of the macromolecules, because architectures or skeletal architectures has an influence on its property as well. So that also we are exploring. So another important class of architectures we are working, that is the macrocycles, because due to its cyclic structure, cyclic nature, macrocycles has its own advantages, own unique properties. We all know erythromycin antibiotic, that's a macrocycles, or the crown ether we know, those all are macrocycles. So we are also developing different classes of macrocycles with different functional groups. For example, one class we can see here, this is a dithiocarbamate based macrocycles, where we can see that via synthetic strategy we can tune its ring structures, we can tune the backbone basically ring structures, the functional groups, and we can also tune the ring structure functional groups and also backbone functional groups as well. So and via tuning these different properties also, we are in this work we have shown that how we can tune its interactions with different proteins. So this is dithiocarbamate. Another class is that we also make the triazole based basically macrocycles, that all the basically we have the similar things, basically ring size functional groups, all the cases we can tune, and also here we can tune the heteroatom in the backbone as well. And in the last slide, I will show that some cases we are also, based on the need, we are also modifying the macrocycles, synthetically modifying macrocycles with a fluorophore to make the fluorescent macrocycles for some applications. So this is the last slide. So I will show two examples. So this is one class of the again macrocycles where we have added the dancyl fluorophore. So they all are the fluorescent macrocycles with tunable ring size and functional groups. And this class it showed selective detection of one explosive, that 2,4,6 trinitrophenol. That is a very common and widely used explosives compound in the explosives, and in the army area all the soil and water, they are contaminated with that one. And this class of the macrocycles, it shows selective detections of this explosive in water as well as soil, and we also have developed the paper strip based sensors. Another example is that this is another class of macrocycles which shows that selective detections of the one class of antibiotics, that is a pharmaceutical sensor. So I will not go into the details of this work. So altogether, in summary, what we can say, that what we are trying to do in our lab is that to design and synthesize different classes of macromolecules, and via functional group and the architectures we are trying to tune its property to make them basically best suited for different applications. And in the application side, we are developing some what I have shown, that sensor as well as removal of different environmental toxic pollutants, and also biological sides. Basically, we are trying to develop the antibacterial drugs as well as to understand what are the factors that are important to interact with different proteins. So with that, I would like to thank you all for your kind attention. And this is our permanent campus. We have moved to the permanent campus completely. So I welcome you all to our permanent campus. So thank you so much.
Thank you, ma'am, for your wonderful presentation. And you have presented another method for the removal or degradation of organic dyes from water. The earlier speaker already mentioned it. So you have presented another method, and I'm sure it will be a future technology for to remove that organic dyes, and also this detoxification of water. Okay. And now the forum is open for discussion.
Very nice, Mintu. Nice to see you. Nice to hear your amazing variety of work. You are becoming a detector woman.
Sorry.
You are becoming a detector woman.
Oh.
Very nice. So what I wanted to ask you was that the very same polymer in its different manifestation, the long chain, the cyclic, the different kind of morphology give you variety. So earlier we had understood that all interactions are functional group specific. But you are coming to this point that their structure, macromolecular structures become important, not necessarily their what monomer structure or functional. Do you have any insight that you are deriving from so that you can extend your structures just as graphene, many different forms? Is it possible for you to extrapolate and understand the problem?
Yes, sir. So that is actually like our goal. Basically, we are starting, as you say, that sequence different polymer is one side, then polymer, then macrocycles. The how we are fundamentally first trying to understand that how the architectures matters for different properties. For example, that when you see the cyclic systems, if we are making exactly comparison of oligomer versus cyclic system, exact comparison. So now the cyclic structures, when it binds with any protein or any target, and we have exactly linear analog, cyclic structure has a less entropy penalty. So that's why cyclic structure has higher binding than the linear flexible structures. Like that, we are trying to understand, and we are also extending like hyper branch type of things, like branching, as you said, the graphite and those things. So that also we are trying to actually develop. So yeah, we are kind of trying to do that also.
Is it possible that you will get greater insights from computations?
Computation, yes, we have started actually using the computational as well, collaborating with the computational to understand structure, like mainly currently we are doing actually linear versus cyclic, that's what, but our goal is to extend. So their functional group and the structures we are trying to understand by DFT calculations.
What is the origin of mercury in Palakkad campus?
I think different industries, industrial work.
No, Palakkad, the campus was not in an industrialized area.
No. Okay. So no, that what we tested in the laboratory, we have taken the water and we did the spike with the mercury.
No, but you said the campus water also you tested.
No, that we have taken the lake and tap water, but we spiked with mercury. So that in the water, whatever the other things are present, in presence of that also it is able to detect. But there we have spiked the mercury.
No mercury there?
No. We did not find mercury there.
Okay, very nice. So the other point is, now that you are a detector woman, you also want to solve it, right?
Yes.
Let me ask you, what is the cost of this polymer?
What
is the cost of your polymer?
That is very less cost, because the starting material we are using.
No, what is it? What is the cost?
I have to check, but it is I have to exactly amount I have to check.
Is it like let us say PP? I mean a polymer, polypropylene or polyisoprene, any such polymer that is commonly used. Is it comparable to that cost?
It is comparable, it is less, because we are using very less expensive like starting material, and in 1 hour we are making a huge scale of the polymer. So it is that we calculated. I don't remember the value. I don't want to say any value that it is not, but it is very less price. Yeah, that is comparable.
Thank you.
Thank you, sir.
The luminescence with mercury, the ligand is getting quenched.
Yes.
Why is it getting quenched?
Okay. So, it's for, so this is actually, so generally if we take the dancyl chloride, the simple where from we start the dancyl chloride is not it's not fluorescent. The reason is that internal charge transfer from this amine to that here, internal charge transfer.
Transfer from the?
From this nitrogen electron reach to that S double bond.
To the sulfonyl
exactly,
SO2.
Exactly. Okay. And when it is attached with any electron donating part, then it gets fluorescence because it hindered the charge transfer. Now, when what happens when we have the mercury or the electron deficient group, then these things they interact with that one, then internal charge transfer again regenerates. So that's what we see the quenching.
And so this does n play a role? N is equal to, you can go longer.
Yes. That plays a vital role for the LO delimiter protection, because
But the efficiency of charge transfer of fluorescence quenching is same for all n 1, 2, 3, 4.
no. When that's what, when we are increasing the number of dithiocarbonate unit, then quenching is more. That means it's interacting more basically. We see that. That's how we see when n is equal to 1 nanomolar range, n is equal to 2, it goes to the picomolar range. Yeah. Thank you.
Thank you.
Great.
Do you have any reason why it is so selective to mercury?
Mercury, yeah. So one general reason is the soft interaction. So basically dithiocarbonate sulfur is soft and mercury is soft, so soft interaction, that is the general reason. But of course that not only mercury, we have the other metals also, they are also big, right? What we have seen is that when dithiocarbonate is a free dithiocarbonate, that means S minus, then it interacts with all the metals. But when it is in the ester form, that's what we make it in a dithiocarbonate ester form, then it is only selective to mercury. So,
Methyl mercury or, only Hg2 plus?
Hg2 plus, that's what we use.
Not monovalent, not to
Actually,
covalent.
we have tested only Ag2 plus. Maybe we have to test the others. Thanks for the suggestions that what happens. We just tested Ag2 plus only. But we'll test those.
So, you can go across Chalia river. Once it was having methyl mercury. Now it is not. But there are other rivers in the country where methyl mercury is there.
Methyl mercury, yes.
And this is a starting point. Start from Farook College.
Sure. Thank you.
It was a nice talk.
Thank you.
So I would like to know, how did you evaluate that hydrophobicity using that HPLC method?
Okay. So in the HPLC, what we use, See is basically it's a column. Okay. What we do, we use two solvents, acetonitrile and water, and we are starting more water, we are decreasing the percentage of water or increasing the percentage of acetonitrile. Okay. And now, here, so that's what basically it is. When it is retention time is slowly it is moving towards, so what is happening is that your water percentage is decreasing. That means hydrophilicity is decreasing and polar thing is decreasing, non-polar things is increasing. Whatever is more hydrophobic, they will come later, they will elute later. Whatever is hydrophilic, they will elute first. Okay, based on that, basically we can see that more hydrophilic compound, whatever comes, it will come first, and more hydrophobic compounds, it will come later. So based on the retention time, that's what we say, when retention time is increasing, hydrophobicity also is increasing.
So can we use this method for any kind of materials?
If it is eluting, basically, it has to come out. Some compounds are like, let's say it's stuck to the column, but generally you can use for general cases actually.
Thank you, ma'am.
Thank you.
Good afternoon, ma'am.
Yes, good afternoon.
Ma'am, I have 3 small doubts.
Yes.
What is meant by tunability?
What is meant by tunability?
Yes, sir.
Oh, very good question. Tunability means like any factor if you take, can you tune it, can you change it, that property, or can you modulate that property? That is the tunability. Let's say you take the same platform and you are changing the properties. That's the tunability. Can you tune the properties? That's the tunability.
Okay, ma'am.
Tune it basically, or change it.
And second question, how dithiocarbamate is applicable in biomedical sciences, having anti-cancer properties specifically?
How dithiocarbamate
is applicable in biomedical sciences, having anti-cancer properties
and
specifically.
I did not show any anti-cancer work.
But there was, in one slide, there was
basically this
mentioned.
group, okay, that's a good question. So there are some drugs, basically in those drugs it is the dithiocarbamate group is present in that drug, and it shows that because of that sulfur, because in the proteins you have the cysteine and different groups, due to the sulfur it specifically interact with those proteins. So that's what they have found out, that dithiocarbamate is important for different basically. It can interact with the biomacromolecules, proteins.
Yes, ma'am. My last question, as was in the last session, Dr. Ujjal sir mentioned about the contamination of dyes in water bodies. So you have mentioned about the removal of organic dyes too. So it's a method applicable to different dyes other than organic.
Sorry, can you repeat that question, last part?
Is the method you suggested applicable to dyes other than organic dyes?
Okay, okay, that's a very good question actually. So of course there are a lot of dyes are present, but we have to test it. In our case, what we are doing actually, the organic dyes where aromatic groups are present, and via our synthetic system, via enriching the aromatic groups, we are trying to develop. So these whatever I have shown, it is only organic dyes. But definitely your question is that that's what, to target those we have to see what are the functional groups are important, and that we have to incorporate in our synthetic system. Then of course that we can actually try to do that. But that's a very good question.
Okay, ma'am. Thank you, ma'am.
Thank you for
Thank you.